Okay, hi everyone. This is chapter 35, Water and Sugar Transport in Plants. Hope you're not tired of plants yet because we still have a whole 3 more chapters discussing plants. So one of the things that we're really going to focus on in this lecture is water and the movement of water. So we're going to be drawing back on some things, some concepts of osmosis and movement of solutes. So we'll review that in this lecture and talk about really how it specifically relates to plants. So these are things you may have seen if you've taken Bio 1. So in order for a plant, specifically a land plant, having to do photosynthesis, dealing with water loss is going to be a serious problem because when a plant takes in carbon dioxide, it has to open up its stomata. And when it opens up its stomata, remember what happens is water is released. So this evaporation or this transpiration, right, another term for it, is going to cause some challenges and some benefits to plants. So it brings a flow of water out of the plant, carrying minerals from the roots and the shoots and cooling the plant. That's a good thing. However, that water loss is going to, if not replaced, cause the plant cells to dry out, wilt, and ultimately die. So we need evaporation. We need water to flow from the roots all the way up through the shoots and out the leaves. That's a necessary flow of water. It's going to move the minerals. It's going to move through our the entire plant body. But like I said, this is not. If this water is not replaced, it's going to be a problem. So all terrestrial plants, the angiosperms, the gymnosperms, everything we've discussed, except for mosses and liverworts, have these vascular systems, right? These vascular plants that move nutrients and water through the plant through tubes of conductive cells. So this is a review. Last time we introduced these, we're going to spend some more time with them today. So the xylem. Remember that the xylem are your dead cells. They form tunnels through which water and minerals move. And the phloem are living cells that carry the products of photosynthesis from the leaves down to other parts of the plant. So the xylem, they go upwards from the roots up to the shoots, and those are dead cells. They're basically dead tubes. And that makes sense if you think about it, right? Because if you picture a tree, we talked about kind of what the internal structure is of the wood and the bark. It would make sense that water would go up through tunnels that were consisting of dead cells. Whereas the phloem, where photosynthesis takes place, those have to be living cells because that's where photosynthesis takes place, and that's going to be a very energy-rich process. So you expect that obviously to happen in those living cells. So xylem, phloem, should recognize those terms. You should be fairly comfortable with them and what they are at this point. So we're going to discuss 3 ways in which this actually happens. So you might be thinking, okay, the water's down in the soil, but it's got to get up to those leaves so that they can use that water, that H2O, to build glucose and combine it with the CO2 and do photosynthesis. So how's that water get up there? Well, there's a few hypotheses that we're going to look at that involve transpiration, adhesion, tension, cohesion. So these are terms that if you've got some chemistry background, you should be familiar with. We're also going to talk about solute potential and its effect on water movement, and also pressure potential and its effect on water movement. So these concepts in tandem will address how water moves. So let's talk about water potential first. So what is water potential? So anytime you think about potential for anything, it's just how likely something is that's going to happen, right? So water potential is how likely it is water is going to move from one area to another. So what's the relative tendency of water to move from an area where there's generally a higher concentration of water molecules to a lower concentration of water molecules? And how do we get higher and lower concentrations of water molecules? Now, we're not going to get into the math of it in this course. Some of you may have at some point. If you've taken, like an AP bio class in high school, you may have gotten into that. But for this course, we're not really going to spend much time. It's more important to me that you understand the concept here, right? So we should understand what a solution is and how that affects the concentration of water. So a solution is a homogeneous mixture of water, let's say, since we're talking about water, and some dissolved substance or a solute. All right, so solutes can be sugar, it could be glucose, it could be anything that you're dissolving in the water that's changing the concentration of water molecules. So you're adding this solute and it's ultimately reducing the concentration of water. So if you have more solute, then you're going to see that more solute is going to change the direction in which water is going to flow, and you're going to have a lower concentration of water. So that's one way in which water moves. That's solute potential. The second way is pressure potential. So you can change the pressure, water pressure, and that's ultimately going to force the flow of water. And water is always going to move, and anything's going to move from an area where you have a high to a low pressure differential. All right. So differences in potential determine the direction of water movement. The net movement of water is from areas of high water potential to areas of low water potential. So when we're talking about plants, the roots have a high water potential, the leaves have a low water potential. So that makes a lot of sense, right? You water right at the roots, high water potential there, high concentration, high pressure of water, low pressure up in those leaves. So where is water naturally going to go? It's going to go from that high concentration to that low concentration. So there's some things to overcome though. There's gravity, but ultimately we're going to be relying on lots of water potential factors that are going to influence that movement of water from the roots up through the shoots to the leaves to ultimately be transpired. So solute potential is the tendency for water to move by osmosis in response to differences in solute concentration. So if we look at this picture here on the right, you may have seen this before, this U-tube, the original U-tube. In this U-tube, we've got a semi-permeable membrane here in the center. So that means that this membrane is going to allow water to flow through, but it's not going to allow these sugars to flow through. So over here we have a high concentration of sugar and therefore a lower concentration of water, relative to this side where we have a low concentration of sugar and a high concentration of water. Based on what we know about solute potential, these water molecules are going to flow from the area where we have a high concentration of water to a low concentration of water. So it's going to move in this direction. And we see that happening here, and we see the water rising on this side. Because what's happening is the glucose, the sugar molecules, are not moving. So they're in green. They're gigantic, right? They're not going to go, but the water molecules are going to flow, and they're going to flow from the area where there's a high concentration to low concentration. Now you may say, wait a minute, there's an arrow going this way. Well, it's because it's a dynamic equilibrium. It's not all going to flow. There is going to be some, but there's going to be a net movement. Okay, in this case to the right, by osmosis. So that's one way, solute potential, in which water will naturally move. And this all happens passively. No energy is needed for this to happen. Okay, pressure potential is essentially the same thing in that we're going to be moving from high to low pressure in terms of water, but there's certain things that affect pressure. So when we're looking at plant cells, wall pressure is the force exerted when the relatively rigid cell wall of a plant resists expansion of the cell volume. So think about it. When water enters a cell here, which is happening, right? More water is in the outside than in the inside of the cell. This cell is going to swell up, and it's going to push on that cell wall. So almost like a water balloon. We call that the wall pressure against the cell wall. Turgor pressure results as incoming water causes the cell to swell and the plasma membrane pushes against the cell wall. So these two things, in my mind, I generally use interchangeably. A positive pressure means a plant is turgid, and a negative pressure means that it is flaccid. So in this case, we're looking at an environment where there is a lower water concentration outside than inside, so water is going to flow out of the cell, and in this case the cell is going to basically pull away from its cell wall. It's going to become flaccid, and this is how we get like wilted leaves, wilted vegetables from these flaccid cell walls. So that's why we want to keep the produce and stuff really plump, so they'll keep it the cells turgid by spraying them down in the supermarket. So how does this affect water movement? So in terms of transpiration, that's when water from the roots to the shoots moves up the plant and is driven by evaporation from the stomata. So in order for transpiration to occur, the stomata have to be open. Okay, that's the only way water is going to go out, is if those stomata, those openings, the leaves are open. And the air surrounding the leaves is drier than the air inside of the leaves, because remember, water is going to move down its pressure gradient from high to low water potential. So we're not going to see this occurring if there's really, really high humidity or if it's raining, right, this is going to happen on a dry day. There's going to be lots and lots of transpiration. All right, so how does this water move up? So one of the ways in which this happens is through something called capillary action, and this is also something that we see happening similarly in the human body, but movement of water up a narrow tube. So example, the xylem element, due to three forces. So these are three forces that have to do with the properties of water. So the first one is adhesion. So adhesion is this tendency for the water molecules to kind of stick together, and when they stick together, that creates an upward pull at the water container surface. Now, in terms of what's happening with our plants, okay, we're going to start the process at the bottom, at the roots, and then this is going to continuously happen and pull the water up. Surface tension creates an upward pull at the top of the water column. So surface tension, an example of that would be, if you've ever seen like one of those, trying to think what they're called. I think it's called actually a Jesus Christ lizard. And what the Jesus Christ lizard does is it kind of can stand on the surface of the water because surface of the water has that surface tension. And it creates sort of this ability for, pull at the top of the water column. So surface tension is another example of something that works with capillary action. In terms of cohesion, transmitting both forces to the water below, this attraction occurs among like molecules such as the hydrogen bonding that occurs in water, and this upward pull by adhesion is transmitted to the rest of the water column. So the water column rises, in this case, against the pull of gravity. So the collective action of adhesion, cohesion, and gravity are responsible for the formation of the surface boundary here of plants, which we're going to see these narrow tubes. So really, in terms of how water moves, it's a collective action of these three things. The forces that exist between water and air and the interference there that's with them. So this is essentially, in so many words, this is a good slide for you to use to study. I'm going to kind of just go through it with you quickly. I'm not going to read every single thing, but keep in mind that water is going to go from the soil to the roots, the stems, the leaves, and ultimately to the air. That's summarized here, and that's what's going to happen ultimately with transpiration. Water molecules that move through the xylem are connected through a continuous stream of water due to the hydrogen bonds. So that's that collective action, like I said, of the adhesion, cohesion, and surface tension, making that upward pull across the surface. And cohesion ultimately transmits both forces to the water below. As water evaporates into the atmosphere, it tugs the adjacent water molecules inside the leaf, which tugs the water molecules in the stem, which tugs from the roots to the soil. So it's this constant sticking together of the molecules to pull it up. And believe it or not, this whole process does not require any energy because it's just dealing with the movement of water forced by the water potential based on pressures and solute potential, which we're going to see a little bit how that works. All right, so here's a good example or a good figure here from the leaves all the way down into the roots. So I know this is number 7 down here, but we know water kind of moves in here, and we know water vapor diffuses out of the stomata. So as this water goes in, it's going to ultimately be pulled up, okay, and then pulled up again at each level of tension. The water is pulled up until it is ultimately released out of the stomata. Okay, so that's, there's going to be some activities you guys do in the mastering that will reinforce that concept because it is a little bit tricky. But if you understand the movement of water through surface tension, adhesion, and cohesion, okay, surface tension pulling water up to minimize that air-water interference, adhesion, water is attracted and pulled up, and cohesion is holding it together. Those three things together will do that. And you can actually, if you put water through a glass tube, you can see this happening. I think there's a picture in your text that shows that as well. All right, so let's talk about roots and roots pressure. So in the roots, we're going to look, and we've seen some of this already when we looked at kind of the growth of plants, but If we start from the outside of the roots in, the very outside is the epidermis, contains the root hairs, and then we've got the cortex, the endodermis, the pericycle, and then ultimately in the deep center of the roots we have vascular tissue, which contains the xylem and the phloem. So here's a look at this, a cross section. Water is going to move obviously down the center where those vascular tissues are, and we're going to look at kind of the movement here, how water gets all the way from here to here, and it's going to have to go through some checkpoints before it ultimately gets there. But you do want to recognize this figure. It's pretty easy. All right, so after water is absorbed through the root hair, so water goes in, it's going to make its way to the tissues, like I said, through these cells from out here to there, 3 different roots. So there's the symplastic root, the transmembrane root, and the apoplastic root. So the names tell you what the root does, not the root R-O-T, but the R-U-T-E root, the root through the root. It's funny, I'm making jokes talking to myself. Not that anybody usually laughs in class anyway, but so water can flow in the symplastic root through the plasmodesmata, which connect these cells together. It can flow through aquaporins the way it does in the transmembrane root. So the aquaporins are literally water pores or openings for water in the cell membrane. Or it can go and actually completely bypass the cell membrane and go around the cell wall, and that is the apoplastic route. So the apoplastic route is a little bit scary because it's literally just water going through on its own devices, not going through any filtration, but there's a gatekeeper for that. So let's look at these 3 roots, and it looks a little bit scary at first, but it's really not bad. So the top root, again, what are we looking at here? Always want to orient ourselves. Here again, water goes in through the root hair, and it goes through the epidermis, the cortex, ultimately getting to the vascular tissue here, the xylem, but it's got to go through all of these cells. So if it goes through and it just goes through the plasmodesmata, it's going through the cells, it's going through the cells, it's going to make its way to the xylem. The transmembrane root, same thing. It's going to go through the cells. It's going to go specifically through the aquaporins. It's going to get filtered out, and things like that are going to happen to it, and it gets to the xylem. Now, what we have going on here is a little bit different in the apoplastic root. So, in the apoplastic root, water is totally not even going near the cells. We don't know what's in that water. Could be any kind of concentration of ions, unregulated. So this structure called the Casparian strip at the apoplastic root is going to basically stop the flow of water so that it's not going necessarily all at the same time to the xylem, and it can be filtered out and go through a different route. So the Casparian strip acts like a gatekeeper. And it's the Casparian strip has a secretion called suberin, which is like a wax that buffers the water. So the Casparian strip allows for water to take an alternative route and controls the flow of water into the cell. Right? So here's that information a little bit more about the Casparian strip. Blocks the apoplastic pathway, and it's made up of suberin, which is a waterproof barrier. In order for water and solutes to reach the vascular tissue, they must move into the cytoplasm of an endodermal cell. So in that case, those endodermal cells act as gatekeepers. So because of the role of the Casparian strip, water and ions must cross at least 2 membranes as they move from the soil to the xylem. One membrane is crossed by water entering the symplast outside of the Casparian strip. The other membrane is crossed by water leaving the symplast outside, and then one leaving inside the Casparian strip. So it adds an extra layer of protection so that plants will use specific channel or carrier proteins to control what moves into the shoots. So the Casparian strip, again, is like the gatekeeper here of what gets into the xylem. We don't want anything nasty getting in there. It's going to harm the plant, pollutants and things like that. Okay, so one of the trade-offs in keeping the stomata open and moving the water again is water loss. Water loss. So if the stomata close to reduce those transpiration rates and stop water from flowing out so frequently, that's going to affect photosynthesis. It's going to slow. It's going to stop. So that presents itself as a compromise, and plants have evolved based on their environment to keep the stomata open and closed at times when it's best for them. Okay, so all of that was water. So all of that was all happening in the xylem. So let's look now at what's happening with the phloem and sugar. So when we're thinking about sugar, remember, sugar is produced in the leaves, and it's got to get all the way down to the plant, ultimately to the roots, to be stored as starch. So translocation is the movement of sugars through a plant by the process of bulk flow. And this occurs from sources where the sugar is made, like the leaves, to sinks where sugar exits the phloem. And sugar concentrations are going to be high in sources and low in sinks. So that makes sense, right? Where the sugar is produced, you're going to see a high concentration of sugar. It's going to be lower where it's going to exit the phloem and be deposited. So source, where the sugar enters the phloem, sink, where the sugar exits the phloem. You can see this is actually going to be different depending on the time of the year as well. So in this picture here, we're looking at the source being the mature leaves, and the sinks being the roots and the flowers, the young leaves as well. So the location of the sources and sinks varies with the times of the year. So early in the growing season, storage cells in the roots and stems are sources, while developing leaves are the sinks, which makes sense. During the growing season, mature leaves and stems are sources, while meristems, remember where the roots are growing, developing leaves, flowers, seeds, fruits, and storage cells in the roots are going to be your sinks. So just keep that distinction in mind between sources and sinks. Okay, so how do sugars move? Well, it has to do with this pressure flow hypothesis. And there's a better picture coming up in a second that I'll really kind of walk you through. But it has to do with the fact that sucrose, a high concentration of sucrose, is going to ultimately pull water from the xylem, and that's going to move our sugar. So sucrose is moved by active transport from source cells through companion cells to sieve tube members. So remember, the phloem these are living cells, and the phloem are going to have these companion cells next to them. So next to all of our phloem, we've got xylem, and water flows passively, flowing from the xylem into the sieve tube elements of the phloem. Turgor pressure is going to build in the sieve tube elements in the source region, and that's ultimately going to pull our sugars. All right, so pressure flow here is showing you number one, step one, sugars are actively transported from source cells. Okay, so here's our source cell. So this could be in the leaves, okay, into tube elements. So in the tube elements, we've got a high concentration of sugar already. This is where we've got like our sap and things like that. So this energy has to be required to pump this sugar out into there. So now we're going to get a really high concentration of sugar in here, even higher than there was before. Okay, so it already was pulling some water out from the xylem over here, but now it's going to pull even more. So H2O water is going to diffuse into the sieve tube elements, raising the water pressure. All right. Pressure causes the sap sugar water to flow through the phloem, okay, and sugars are transported out of the phloem into sink cells. All right, so we're going to see water flowing out here, okay, ultimately, as it flows through, sugars are going to be passively moving here into our sink cells where there already was a low concentration. Okay, so this is all going to be phloem loading, so loading the phloem with sugar. What's happening down here is phloem unloading. So cells in the sink remove sucrose from the phloem sap by passive or active transport. So usually it's going to be passive because there's already a concentration gradient there, high solute to low solute, but sometimes more is pumped out. But due to the loss of solutes, water flows passively and turgor pressure in the sieve tube elements drop. The The net result of phloem loading and unloading is high turgor pressure near the source, low turgor pressure near the sink, which is going to drive that phloem sap from the source to the sink via bulk flow. So bulk flow is just the movement of everything all together. There's a one-way flow of sucrose and a continuous loop of water movement as water flows between xylem and these sieve tube elements. All right, so again, here's a better picture. This is the one directly from your text. And you can see nice, even more clearly here, your source cell. You got the sucrose, which is going to be actively transported here into the companion cell. So we got a low concentration here, and we're going to push it into a higher concentration, and we're going to add even more sugar to our phloem. What that does is it draws water out of the xylem, okay, and through pressure, flow, and bulk flow here, the sucrose is going to move out of the phloem to the companion cell to the sink, and the water is going to move back out into the xylem and go up the plant. So it's essentially like the phloem is hijacking some of the water from the xylem to push sucrose more quickly into the sink cells, in this case the root. So how does this all work? Some of this is a review, again, probably of Bio 1. But there's two types of membrane proteins that are responsible for this movement. So you'll notice there's these little channels here. And these are all membrane proteins where these things are going to move. And some of them are open to sugar, and some of them closed to sugar, some of them open to water, closed to water, and that's how this whole thing is driven as well. Channel proteins specifically are going to selectively admit ions and literally just move right through like a channel. Carrier proteins undergo a conformational change to transport specific molecules across the membrane. So these carrier proteins are like a lock and key mechanism. So when a specific molecule locks into the carrier protein, it's going to change its shape, it's going to open up, and it's going to let it in on the other side. So I'm kind of making, you can't see me with my hands, I'm making like an open gesture, and then the thing's going in, and I'm opening up on the other side. That's what happens with a carrier protein, whereas a channel protein it zips right through. We're talking about moving ions or molecules against their concentration gradient, or the electrochemical gradient, from a low to a high concentration. That's active transport, and that's going to require the cell to input energy. Whereas osmosis and diffusion, moving from high to low, those are all examples of passive transport. Okay. All right, so that will end chapter 35 on water and sugar transport in plants.